Virtual and Personality Health Network Virtual Brown Bag Series. Today we are very fortunate to have two speakers that will be discussing the field of health neuroscience. So our first speaker is Pete Gianaros, and he's a professor of psychology and psychiatry at the University of Pittsburgh, and he's also the director of the Behavioral Neurophysiology Lab. And we also have David Creswell with us. He's an associate professor of psychology at Carnegie Mellon, and he's the director of the Health and Human Performance Laboratory. And we're very fortunate to have both of these speakers because they've really helped to pioneer the field of health neuroscience and are going to be talking to us about this field today. So during the talk, I do encourage everyone to ask questions by using the chat function on your screen. If you look towards the right-hand side of your screen, you'll see a little window that says chat, and you can use that window to ask questions of the speakers. So you will direct your questions to David Creswell, and he'll be able to see those questions and then either answer them during the talk itself or perhaps wait till the question and answer period at the end to answer those. So I'm going to go ahead and welcome our two speakers and turn the audio to them. Welcome both Pete and David.
Okay, great. Thank you, Lisa, for that introduction, and also thank you for inviting us both to be doing this. It's our pleasure to talk to you about health neuroscience. Our goal for today was to talk a little bit about what this field looks like, what kinds of questions we ask in this area of research, and also we'll talk a little bit about where we see the future of this field going. Before we talk about health neuroscience, we're going to talk a little bit about our backgrounds and how we got into this area of research, and then we'll go through the slides. But we wanted to emphasize that we were hoping to make this as interactive as possible. So please do feel free to use the chat function and ask us questions as we go along. We're more than willing, and we're very happy to take questions and make this as conversational as possible. So I could start with myself. As Lisa mentioned, I'm a professor of psychology and psychiatry at the University of Pittsburgh. I was trained in a psychology department as a psychophysiologist, and I began my research in the area of stress physiology as it relates to risk for heart disease. And over about the last 10 or 15 years, I've been doing more research that is focused on understanding how the brain generates patterns of stress physiology that might put people at risk for heart disease. And in other work, I've been trying to understand the neurobiology of social health disparities with a particular focus on how socioeconomic status might adversely affect the brain and some of the mechanisms that might be involved in how different features of socioeconomic status might relate to brain health over the lifespan. That's really, I think that's probably sufficient to get us going. I'll turn it over to David now. He can tell you a little bit about himself.
Yeah, and this is our first time doing something together and via webinar. So you know, we're apologizing up front as we try to figure this out. Keep trying to remind myself to stare at the camera. And I see we have a number of attendees already, so that's great. We hope that we can make this you know, interesting to you guys, and probably our family members that are on there right now. So I'm David Creswell. I'm a professor, associate professor in psychology here at Carnegie Mellon University. So we're both in Pittsburgh. We actually have a wonderful group of people that I think are trying to advance health neuroscience research here in Pittsburgh. So it's a real pleasure for us to be able to sit in one room, I think, and do this webinar. So broadly, research in my lab focuses on trying to understand you know, what makes people resilient under stress, and certainly sort of taking the lead from work that Pete's doing in terms of understanding the neurobiology of stress in humans, we've been trying to look at this in a different way and sort of say, lots of people face stressors, and not everybody gets sick. So what are these sort of underlying neurobiological and physiological mechanisms underlying resilience? And so we work there. Most of the work that we do sort of takes an experimental approach where we really use stress management strategies or interventions as an approach for a probe for sort of studying resilience. And so some of the work we do right now, a lot of the work we're doing right now is on mindfulness interventions, and I'm going to give you at least a snapshot on some health neuroscience work that we're doing there. And I think that's it.
David is an amazing golfer. All right, so with that, I thought what we could do now is just launch into a discussion of health neuroscience. And here what we're going to do is describe some of its major themes and some of the assumptions that we've made moving forward. And then we'll give you some illustrations of some kinds of studies and what questions look like in this field. And then we'll talk about its future. But again, if at any point questions come up or you want us to elaborate on something, give more examples, we're happy to field those questions as we go along. So health neuroscience, the simplest way to put it is that it's an interdisciplinary field of research that's at the interface of health psychology and neuroscience. And as we see it, it's a field that's primarily concerned with how the brain influences physical health and how physical health influences the brain, again, very broadly stated. And a lot of the ideas that we'll be talking about in the next few slides are articulated in a paper that David and I wrote with our colleagues here at Pitt and CMU, Kirk Erickson and Tim Versteinen. And this paper is publicly available in PubMed, and we have a link to it down here at the bottom of this slide. So a lot of what we're going to talk about comes from this paper, and which goes into a little bit more depth if you're interested. So as we see it, health neuroscience is subsumed broadly within health psychology. But it's a little bit different than standard health psychology insofar as we have a primary methodological and conceptual focus on the brain. And we'll elaborate a little bit on what that means a little bit later. It's slightly different from neuroscience as a big field, or cognitive neuroscience or social neuroscience. In that the primary focus, as we see it in this field, is on physical health, as well as psychosocial and behavioral factors that influence physical health. So there are lots of fields of neuroscience, for example, that are interested in physical health, but here, because of our additional focus on psychological, social, and behavioral processes, we see this as slightly different than other fields of neuroscience per se. Some common themes in this area of research. So basically topics that you would see illustrated in papers that are published in this field. These themes would focus on brain and behavior relationships and brain and physiology relationships that are specifically determinants or markers or consequences of physical health and illness. And we'll give you to make this concrete, we'll give you some illustrations of what these kinds of relationships look like as they relate to, determining or serving as a proxy for health, or being a proxy or a consequence of physical health and illness across the lifespan. One assumption that we've made in this area is that doing the kind of work, doing health neuroscience research, will give us a better understanding of how it is that the brain comes to link genetic factors, biological, behavioral, social, and other kinds of factors that are of interest in health psychology with physical health and vulnerability to illness. So in this sense, we see health neuroscience as advancing mechanistic understandings of topics that are of interest in health psychology. And another premise is that, by doing this mechanistic work, it may be the case that we can develop like new platforms to develop neurobiologically informed or brain-based prevention and intervention efforts that are geared toward improving physical health and promoting well-being as well, which is something that, again, sort of differentiates, I think, this field from the broader field of health psychology, insofar as we're bringing tools to bear that focus on understanding brain mechanisms and using brain-related information to inform efforts that can promote health across the lifespan.
So I want to just jump in. I think on this idea around what's the added value with these sort of brain-based approaches, I think there's both basic value in terms of building a better mechanistic understanding of how the brain mediates a lot of the things we care about in health psychology. And I think Herb Weiner, in one of his books, has this quote that I sometimes talk about, where he says, we know a whole lot about how stress can influence disease, but the amount we know about how the brain sort of drives these processes is so underdeveloped. And so I think a lot of what we're doing with health neuroscience is trying to build out a better understanding of what the brain is doing in terms of mediating or otherwise affecting these processes we care about so much in health psychology in terms of how psychosocial factors influence health. So this idea around building new platforms, there's other added values. We were sort of brainstorming about this, I don't know, some time ago, about how there's a lot in terms of neurostimulatory devices and neurofeedback approaches that I think offer ways for us to sort of use the brain in terms of changing health over time. And I don't know, I can only think of one or two studies in these domains that I think is so. I mean, it's really wide open.
Yeah. Yeah, and we can come back to that, and as we field questions about where you guys see this work fitting in, moving forward from a more applied perspective as well. Okay, so there's no questions, which means that all of this is perfectly clear. Okay, so this is a figure from the paper that I referenced earlier that puts in graphical form a lot of the ideas that I just summarized, as well as a couple of other ones. So again, health neuroscience is at the interface of health psychology and neuroscience, and it sort of positions the brain in a central location. And some of the main ideas that are being conveyed here are as follows. So one is that we assume that the brain links social, cultural, environmental, and other influences on health. So in that sense, the brain is a central mediator of a lot of the psychological, social, and other factors that the health psychologists are interested in. So from that mediational framework, you can then link out to health behaviors, patterns of physiology, like stress physiology, that are orchestrated by the brain that in turn influence well-being and illness. So in that sense, you can consider the brain as a determinant or sort of the orchestrator of these factors and processes like cognitive functioning, stress and emotion, health behaviors, and so forth. So here the brain is a mediator, and it's a determinant via these top-down pathways of these phenomena that influence health and well-being. Another assumption is that a lot of the relationships that we're interested in health psychology are bidirectional insofar as the brain is concerned. So for example, patterns of stress physiology, health behaviors like, for example, smoking, sleep, and diet can also come to affect aspects of brain function and structure as well. So these bottom-up pathways consider the brain as an outcome variable in a sense, insofar as it's the target of a lot of these processes. And so, for example, I'll give you some examples of what treating the brain as an outcome variable might look like in this scenario a little bit later. But the take-home message is that the brain is a mediator between a lot of these influences on health. the brain can be a determinant of these downstream processes, and it can also be a target of these processes as well. I don't think that there are other points. We'll return to a point later. We're making a couple of assumptions, and we recognize some arbitrary limitations of how we're conceptualizing health and well-being, but we can return to those a little bit later. But I just wanted to run through some of the takeaway messages from this figure that, again, sort of illustrates the ideas we talked about earlier, and typifies some of the assumptions that we're making. Just as an aside, all of this actually did start in a bar over drinks with Kirk and Tim, and up over here on the left-hand side, I'm showing you the doodle drawing on a piece of scrap paper that led to this figure and a lot of the ideas that are in this figure. And yes, this says "cool picture of brain" in the center, which oh sorry, that messed up. And the cool picture of the brain turned out to be this thing down here. So anyway, I knew David would like that. Okay, all right. So before we get into the illustrations and examples of studies from this area, a couple of other points we wanted to make are that the methods in this area are definitely multi-level. So for example, you can see studies in health neuroscience that go from community-level SCS using census-based information down to the level of the brain, and then down to the level of cellular function as well. A lot of tools get integrated in this field. I'll give you an example of a machine learning study of neural correlates of stress physiology in a second. But a lot of tools get integrated, which poses a lot of challenges to doing team-based science, and also poses a lot of challenges in terms of what you're going to see with big data problems, as well as training issues. So it's something that we can come back to later. Is if you're a postdoc interested in getting into this area and you have zero background in neuroscience, it can oftentimes pose great challenges that we can talk about a little bit later.
When Pete, you came into a postdoc sort of re-specializing in neuroscience. This can be done.
Yeah, it can be done. It's not like big walls, but I think it poses challenges on how to tailor your training to make sure you can address the questions that you want to address. Okay, so then the last point that we'll make is that a lot of the questions that you see addressed in this field are the same as you see in health psychology. So if you pick up any standard health psychology textbook like Shelley Taylor's, every chapter probably has some kind of representation in a neuroscience study now, and I haven't gone back and looked at this, but a lot of the questions concern what are the neural pathways by which stress-related factors influence health? What are interactions between the brain and stress-related physiological pathways concerning autonomic function, neuroendocrine function, immune interactions, brain-gene interactions in the context of risk for chronic illnesses, and so forth. There are a lot of studies on these topics, and there are people who we admire a lot who are doing great work on these topics. In the interest of time, we can only probably give you illustrations of some of these, and David and I are going to be wringing our hands about people we forgot to mention or studies we forgot to give a shout out to. So please pardon us in advance. Okay. We have one question right here so far. So let's see. To what degree do you think?
Hey, Mary Frances. Hi, Mary Frances.
So Mary Frances is asking us about, when you're doing functional brain imaging, what goes into the choice of what kind of task that you use. I can actually address this one. Why don't I start working through an example, and then I can address this question, Mary Frances, and I'll repeat it for those of you who are listening
I've got some examples too. And
yeah.
Those I'm going to talk about.
Yeah, great. Okay, so is this is clouding their screen probably, right? Because we're sharing the screen?
I think so.
Yeah. Oh, wow. Okay. All right. Page down. All right, so just to start giving you a flavor of what some studies might look like in this area, we'll talk about some of the ones that we know best, including work from our own research. So as I mentioned at the beginning of the brown bag, my primary area of interest is on stress-related physiology and how the brain generates patterns of stress physiology and represents these changes in the context of risk for heart disease. So a lot of my own work uses fMRI to try to identify neural correlates of peripheral changes in blood pressure and heart rate that are evoked by psychological stressors and that have been linked to risk for heart disease. So as many of you probably know, people who have a tendency to show large and prolonged rises in blood pressure and heart rate when they encounter a stressor are at risk for going on to develop hypertension, having a stroke, having a heart attack, and showing an accelerated progression of heart disease, as well as dying early from heart disease. So a lot of the questions that I have are about, well, how is it, what are the brain systems that generate these responses, and how does the brain regulate these responses and terminate them, and so forth. So to do that kind of work, we bring people into an fMRI session where we look at brain activity and peripheral cardiovascular physiology simultaneously while people are engaging in stressor tasks that we've modeled from epidemiological studies. So a lot of these tasks you're familiar with. They involve exerting a lot of mental effort during tasks that are frustrating, that involve a lot of negative feedback, and performing under time pressure. So in that way, we can look at brain activity and peripheral physiology simultaneously to understand, what's going on in the brain while these stress responses are unfolding. In work that I'm finishing up right now, we've imaged people during stress tasks, and then we've assessed them for cardiovascular risk, and we've tracked them over several years. And right now, we're trying to use stress-related brain activity to predict the multi-year progression of preclinical heart disease. So Mary Frances O'Connor, who we hold in high regard, has asked us a question about how we choose, for example, stressor tasks in this kind of work, or tasks that might be linked to some other kind of factor that you're interested in that models a process that's relevant to understanding disease risk. So how we chose our tasks was basically informed by health psychology and behavioral medicine laboratory work that had focused on cardiovascular reactivity. So for example, there's a famous epidemiological study called the Kuopio Ischemic Heart Disease Study that involved trying to understand psychological and behavioral and biological risk factors for heart disease. And as part of that study, they used mental stressors. One was a modified version of a Stroop color word interference task that elicited reliable individual differences in stress-related changes in blood pressure and heart rate. So we took that task as it was performed in the laboratory, and we modified it so that you could implement it in an fMRI scanning paradigm. And our reasoning there was that it would basically provide for some parallels and cross-linkages with an epidemiological and behavioral medicine literature. It also is a task that we thought was important because it elicited reliable individual differences. So we did work where we basically scanned people on multiple occasions using the same task, and we looked for stability of neural responses to the task over time to make sure that we were indeed assessing a reliable individual difference or trait-like factor. So I think a lot of the times the choice of your task is should be informed by prior research, and when you're doing new imaging work, trying to make concrete linkages to prior work that's been done in health psychology or behavioral medicine, so that people, in many respects, so that people without a background in neuroscience can have some sort of like toe-in or understanding of what it is that you're doing. So it's like, oh yeah, I know the Stroop task, I know it elicits heart rate reactions and so forth. So it allows a little bit more of a buy-in.
Yeah, I want to just add to that I think we as health psychologists and social health psychologists we have a repertoire of sort of tasks and manipulations and approaches that we know in our literature, and I think the neurosciences and health neuroscience, for that matter, can really benefit from that. And we've had fun in some of our studies, kind of similar to what you did, in terms of figuring out, well, how can we make this task suitable for an fMRI environment? And so, we've been doing some work on self-affirmation processes, which has been around in social psychology for a couple decades. And how do you get people to sort of systematically think about important personal values, and to do that in the scanner environment? It was actually a lot of fun, and it was something that Janine Dutcher, who I've been working with for a while, has been working on. And to the degree that we can find ways to make these bridges from health psychology of behavioral medicine or from social psychology to the neurosciences, I think can be a real added value, because it starts to provide bridges between literatures that have largely gone,
existed
in parallel.
Yeah. So I just, to echo what David said, I think our advice is to basically look in the field, see what's been done, and then try to adapt it. And there's lots of illustrations of this. So for example, Torwager has done work on a speech preparation task that looks a little bit like a Trier social stressor. If you're interested in modeling processes like social rejection, Naomi Eisenberger and others have used tasks to model those kinds of processes as well. So think about what you're trying to model and what exists out there already. Yeah, we can come back to that a little bit later too. So here's just sort of a high-level example of the kind of work that we do. This is a study where we were trying to, again, identify neural correlates of individual differences in stress-related cardiovascular reactions. So we had about 320 people complete a modified battery of stressors, including the Stroop color word interference task, while we looked at cardiovascular physiology and brain activity at the same time. And we used machine learning to train basically a pattern of brain activity that predicted individual differences in blood pressure reactivity to the tasks. And then, so we developed a pattern and then tested it in an entirely new sample that wasn't used for training. So basically what we were left with was a whole brain pattern that you could compare individuals to, and the extent to which they conform to the pattern, the more likely it is that they were to show a larger response in blood pressure to these tasks. What this kind of study illustrates is how you can integrate methods like machine learning with standard behavioral medicine and imaging approaches to again identify what are the brain systems that are involved in generating stress responses that put people at risk for heart disease. Using tools like machine learning also allows you to address a lot of the problems that are inherent to doing imaging studies. So for example, overfitting, addressing issues of reliability, and so forth. And so this work will be appearing in Journal of the American Heart Association in a little bit if you guys are interested in learning a little bit more about this sort of integrated approach. I would also, to go back to the figure that I showed a little bit earlier, this would be an illustration of a kind of study that's interested in a brain as determinant or top-down pathways, in the sense that we're trying to go from psychological stress to brain activity to downstream patterns of physiology that are controlled by the brain that put people at risk for heart disease. So this would be an example of sort of a top-down approach in health neuroscience. This is a schematic figure summarizing some findings that were published a little bit of time ago. So in this study, we're interested in some of the biological and behavioral pathways that linked different aspects of socioeconomic disadvantage to the integrity of white matter tracts in the brain. So here we use mediation modeling to look at, to ask the question of whether factors like smoking, central adiposity as measured by waist circumference, as well as systemic inflammation as measured by C-reactive protein, served as mediators that linked different dimensions of SES, like low education, low income, and living in a disadvantaged community, to the integrity of white matter pathways. So in this work, we found that people with lower levels of education smoked more, and then this accounted for lower levels of white matter integrity throughout the entire brain. And this kind of work is based again on a confluence of research that had separately looked at socioeconomic disadvantage and risk factors for chronic illness, like smoking, adiposity, and systemic inflammation. And then a separate literature that had looked at things like systemic inflammation and white matter integrity in the brain. So this is an illustration of how to pull sort of two lines of research together to address integrative questions in this field. And this would be an example of a brain as a target or a bottom-up kind of pathway study, in the sense that we're going from factors like smoking to inflammation to central adiposity to predicting the brain outcome measures like white matter and integrity. So here the brain is a target, and then we're also bringing in some of the social and contextual factors that might account or modify some of these relationships.
So Keely's asking a question about this challenge that we have around recruiting larger samples for these types of studies that require you to sort of work across levels or look at individual differences and the costs. The sort of, challenge with how do you get bigger samples at the same time, knowing that imaging on average is costing anywhere from what $400 to $700 an hour to use the scanner. I know your studies have used you sort of gone big.
Yeah. So the question is, imaging is expensive, and if you're interested in individual differences, like most social and health psychologists are, how do you do this? So the one approach is to just grind away at getting grants to do large N studies. Another approach is to try to connect with people who have data sets that you can do secondary data analyses on. And also there's, one of the things that we like to see is the increasing availability of public domain data sets where you can ask a lot of health neuroscience questions. There are, a lot of individual difference variables that get released along with the imaging data that allow you to sort of probe these questions. There are a number of child development, adolescent development studies coming online as well. So, it's grind away at grant applications, connect with a colleague who has data, tack on, try to do an add-on of imaging to ongoing like epidemiological kinds of work, often works as well. Or trying to take advantage of publicly available data. Those are the three, I think, most straightforward things, but it's always a struggle.
I will say, though, so a couple of years ago, I was not getting health neuroscience grants funded, and I sat down with you, Pete. I don't know if you remember, and you gave me some good advice and sort of said, take the long view with this work, and that you may early on not be able to do the big N types of studies, but you can, do reasonably powered smaller studies that allow you to, in an incremental way, sort of build a foundation, so that you're going to be, able to land the R01s to fund much larger trials. So I agree with all your points around how to, leverage existing data sets and, partner up and do those types of things. But I do think that if you do a good job of selecting the right functional tasks and looking at the right health outcomes and the right at-risk populations, those things are all going to boost your ability to detect effects. And, you can then leverage those effects into grant applications or into publications. So, it's a challenge, particularly if you're, wanting to do, individual difference types of studies or causal chain analysis or mediator types of studies where you do need more power for these things. But a good idea can go a long ways, in that you have to sort of take the long view and know that there are ways to leverage smaller N studies to be able to do your dream studies.
Yeah, and to take what David's saying, when he got here, David invested a lot of effort into basically establishing the infrastructure to do intervention studies. So what he spent a lot of his time on doing was basically getting access to participants and establishing relationships in the community where he could do his interventions, and then having a handful of high quality, basically like proof of concept studies that basically allowed him to swing for the fence with larger grants and do the kind of work that he's doing now. So yeah, I agree. You have to start with something manageable, establish the infrastructure, and take the long view, like David is saying. So it's a time investment, but it's a marathon. Okay, so disadvantage and health. And so those are just a couple of examples of brain as a mediator, brain as a determinant, as well as brain as a target. And like we said earlier, David and I think really highly of a lot of people who are doing great work in health neuroscience, and there's lots of examples that we could have chosen. So for example, like Naomi Eisenberger and Tristan Inagaki and Keely Muscatel are doing great work on social factors and some of the mechanisms by which social processes influence health from a neuroscience perspective. James Cohen is doing work on social factors and how processes like social support might buffer stress physiology at the level of the brain. And there's other great work from more of a bottom-up perspective on how patterns of peripheral physiology can come to influence how the brain encodes environmental information and appraises emotional and other kinds of factors. So these are just a couple of highlights among many. Mary Frances O'Connor is doing great work on some of the neuroinflammatory pathways that link grief with health outcomes as well. So I'm going to stop talking for a little bit here and turn it over to David, who's going to talk a little bit more about the health behavior sides of things and also some of the intervention work that's being done in this area.
Yeah, as we do this sort of high level, we're sort of taking more of a forest approach here and picking out a couple of trees and doing some highlights. So I feel like we're not doing the field justice, because I think there's so many exciting areas. I will say for trainees, particularly trainees in the social health area, the Social Personality Health Network's got a lot of sort of social health people, social psychologists working on health-related questions that, I think health neuroscience has got such a huge upside for the coming decades. We have with the advent of bold fMRI in the mid-90s, and then really over the last 10 or 15 years, just the acceleration in terms of the number of ways you can exploit bold fMRI or other types of neuroimaging approaches. There's just so many opportunities for people, I think, to make huge advances in this area. So I hope maybe we're inspiring some excitement for folks to work on this. Probably should save that comment for the end. But as P mentioned, I wanted to say just a little bit about health behaviors, health behavior change. I know these are areas that are of real interest to social psychologists working in the health domain. These are just a couple of highlights. Certainly Elliot Berkman and Emily Falk and others have been doing a lot of work trying to take the very well-established health behavior change literature really in behavioral medicine and social psychology, and trying to understand how the brain mediates these types of processes, and that may add added value above and beyond what we already do as social psychologists. So I put up a nice paper here that Elliot Berkman did, came out in Psych Science now a number of years ago, where they were looking at smokers. And certainly one of the areas that we care about is getting smokers to quit. And Elliot and Emily have been working a lot in terms of understanding brain mechanisms underlying effective health behavior change in the context of smoking. They've worked in other areas as well. But just to orient you to this work, so what they've done in these studies, or at least in a couple of these studies, is they take people who are signed up for smoking cessation programs. So these are typically really heavy smokers who want to quit. And in this particular study, what Elliot did was, after they had started the smoking cessation program, he brought them in and had them do a scan session where one of the things he looked at was their sort of response inhibition response. And in this case, he had folks do a go, no-go task, popular task in cognitive neuroscience, to sort of look at your ability to sort of withhold a sort of prepotent response. And so the idea here with this type of response inhibition task, well, there's lots of go, no-go tasks, but this particular one, you see letters flashed up on a screen, and for most of the letters, it's a go response, so you press a button in response to that go stimulus. And then on a small number of trials, it's a no-go trial, you'll see something like an X or some other type of letter that's your no-go response, and you're supposed to withhold a response. So it's an interesting task in that I think the theory is that a smoker might have a craving response, and their sort of prepotent response is to reach for a cigarette to satisfy that craving. And to the degree you can sort of inhibit that kind of motor response, that desire to act on that craving response, might be a good predictor of who's going to be successful at quitting smoking over time. And so they use this response inhibition task as a way to sort of model how the brain might be able to regulate one's response under high craving states. And the idea here is that maybe it's the case that if you can have a better kind of response inhibition to this task, might be a better predictor of who's going to be able to quit smoking in these smoking cessation types of programs. So they did this response inhibition task, then they used experience sampling. Basically, I think it was a day before they had agreed to quit smoking, and basically measured their smoking behavior in daily life as well as their craving responses. And then they did, at the end of the study, measure, they sort of biochemically verified who had effectively quit using this sort of carbon monoxide approach that's fairly common in the smoking literature. And what they find is that, first of all, they get a reliable neural response in areas that are known to be important in this type of go, no-go task and response inhibition. So they saw activity in the basal ganglia, the pre-supplementary motor area, and the inferior frontal gyrus. So they got a nice sort of neural response to this. And the question was, is this neural response going to moderate the sort of established association between having a daily craving and that making you want to reach for a cigarette and actually smoke? As well as, is this brain response going to predict who is actually successful at quitting by the end of the study? And indeed, what you can see in the bottom right panel here of this slide is that folks who had stronger brain response in these areas that we know are important in response inhibition, in particular in the right inferior frontal gyrus. This is an area that we know is important in sort of self-regulatory responses and across a number of tasks, including this type of response inhibition task. But essentially, this folks who had stronger right inferior frontal activation to this response inhibition task early on in the study had basically severed the link between the degree to which craving was making people grab for cigarettes in those following weeks. So folks who are showing a greater response inhibition response are able to kind of eliminate that craving to behavior link where you experience craving and you then reach for cigarettes. Moreover, they did find some evidence that basal ganglia activation to this response inhibition task did predict successful smoking cessation at the end of the trial. So some initial evidence here that we can use the brain in ways to understand mechanisms underlying health behavior change, in this case smoking behavior. So a nice sort of early proof of concept study around how response inhibition tasks, and in this case what Elliot did was sort of adopt, I think, standard tests that were used in the cognitive neuroscience literature and affective neuroscience literatures, and sort of bring those over to health psychology as a way to leverage understanding health behavior change processes. This group also did some work a couple years ago looking at quitting smoking in a different way. In this case, they were looking at, this was Emily was the lead author in this work, and she's since done a number of studies like this, looking at how smoking ads might affect the brain in ways that might matter for effective behavior change. And so what they did was they brought, again, folks who were in smoking cessation programs, heavy smokers that were in smoking cessation programs, who really wanted to quit, and they showed them in the scanner smoking ads, or stop smoking ads. Let's see if this works. This could backfire. Let me try it this way. Okay, hopefully you can see that. Does it work?
Steve, are you smoking?
Yeah,
Didn't we agree you'd go outside?
it's raining.
I don't see why you can't just quit.
Do you think I've tried? I don't know what else I can do.
You're a finger, Steve. You can dial 1-800-QUIT-NOW for free information and support. Please, Stevie. Quitting is hard, but dialing 1-800-QUIT-NOW is easy. If you're ready to try, call for free information and one-on-one counseling.
So they saw, hopefully you can see that, okay? Is he going to play something else? Okay, hold on. Technical difficulties. Okay. So these smokers who wanted to quit smoking came in and during the scan session viewed short videos like this. They measured their brain response. And they're particularly interested in medial prefrontal cortex, an area that's important in sort of self-regulation, thinking about the self, among other things, and that Emily has now shown in a number of studies to be important in sort of driving health behavior change effects. So they're particularly interested in this area of medial prefrontal cortex. What they find is that when they measure by self-report a broad range of factors that we know are important in behavior change. So in the top right panel here, we're sort of showing you her figure 2 from this paper. But they measured, again, this was early on, their intentions to quit, their beliefs in their ability or self-efficacy to quit, these types of factors. And then they measured whether they predicted the effectiveness of their behavior change. In this case, they took, again, carbon monoxide measures at baseline and then at the end of the study. And you can sort of create a change score to evaluate who's changing the most in terms of reducing their smoking. And they find that self-report does predict behavior change in the expected direction. So the degree to which you have these intentions and beliefs, they are going to, and by self-report, they are going to predict improvements in response to the smoking cessation program. But critically, when they look at this sort of marker of neural activity, in this case, medial prefrontal activation in response to these short ads about quitting smoking, they find that in hierarchical linear regressions that it predicts increased or explains increased variance in terms of your ability to reduce your smoking intake above and beyond self-report. In fact, as you can see in the figure caption here, what they say is that these self-report measures that we typically look at in the health behavior change literature are predicting about 15% of the variance in behavior change. But when you add the medial prefrontal activation in response to these smoking ads, you now are explaining 35% of the variance in behavior change, a significant increase in variance explained. So it does seem like these neural markers can add to our ability to identify who's going to be effective in terms of changing their health behaviors over time. So just 2 snapshots here that I've presented around interesting health neuroscience work on understanding health behaviors. In social and health psychology, we care about a lot of other health behaviors, whether it's getting screenings, mammography screenings, using suntan lotion, eating more fruits and vegetables, and so on. And there's just been very little work in health neuroscience in terms of understanding how the brain mediates these processes and may add added predictive value to the existing models. So hopefully that will get some of you health behavior folks out there interested in working in this area. So before we sort of turn to kind of broader considerations in the field, I wanted to close with a couple of comments about interventions. Interventions have been really a bedrock of health psychology research, going all the way back, behavioral medicine research as well. Yet we know very little about how these interventions change the brain in ways that are going to mediate health outcomes over time. And so we've been working on this, and you can sort of talk about interventions kind of in a broad heuristic way around mind-body interventions, where we're sort of targeting training and psychological processes in ways that are going to change the body, change physiology, change health and disease over time. And so I put mindfulness interventions in this category. We could also talk about body-mind interventions. I'll mention one example of that on the next slide. But my lab, we've been interested a lot in how these mindfulness interventions really become biologically embedded, how they are changing the brain and our peripheral physiology in ways that are going to help explain why we see in a number of randomized controlled trials that mindfulness interventions can improve health outcomes in a broad range of patient populations. So we've been trying to do some of this health neuroscience work. I put up one of our intervention slides here. This was a study we did with stressed unemployed adults in Pittsburgh. I took Pete's advice and used my startup funds to do this initial trial. Blew in my entire startup package, in fact, to do this trial, but it was worth it because it was something that I really believed in and I thought would be a foundation for building future studies. And so what we did in this study, we brought stressed out, unemployed, job-seeking individuals in the community, and they did a baseline appointment. We brought them in and did an hour-long fMRI session, both before and after the mindfulness or control intervention. We did a number of things. We're still just sort of cracking the lid in terms of looking into the different tasks that we used in this particular study. But one of the tasks that we used that's borne a lot of fruit was looking at resting state functional connectivity. So in the absence of any task, we can have people just stare at a fixation cross and sort of look at how, in this case, mindfulness training might be influencing kind of functional connectivity of brain regions at rest. And the default mode network comes up a lot as sort of an interesting target for looking at resting state functional connectivity. Lots of debate in the neurosciences around what it means in terms of the activity and functional connectivity, but certainly it allows us to probe the resting brain in interesting ways. So we did do a resting state scan in the study. After they came in for this baseline fMRI session, we then invited them to a retreat center where we took a blood sample. We wanted to look at these circulating markers of inflammation, which we know are elevated in high stress individuals such as the unemployed. Once they're at the retreat center, we randomized them to one of two wings of the retreat center. One wing they went to was, if you're randomized to the mindfulness program, we called it the Health Enhancement Through Mindfulness Program, or HIM program, and you spent 3 days doing mindfulness-related activities. If you got randomized to the relaxation program, we called this the Health Enhancement Through Relaxation Program, or HER program. You went to the other side of the retreat center where you were doing guided relaxation exercises that we had specifically designed not to sort of foster these mindfulness skills. After they completed we did do some Palm Pilot experience sampling assessments during the retreat. After they did the retreat experience, in the weeks following that, we invited them back to campus again. We had them again complete another fMRI session where we did do resting state functional connectivity, so we could sort of look at how mindfulness training might be modulating this resting state functional connectivity signal. And then we did bring folks in at 4-month follow-up where they again provided a blood sample so we could look at these circulating markers of inflammation, as well as how they liked the program. I will say a couple of things. One is that this approach really worked well. We know high-stress populations typically drop out of interventions. At a retreat, we had them all captive, so they stayed in the program. We had really good retention there. And these unemployed folks were a really unique population, great for studying stress processes with interventions because they had lots of time on their hands. They they really didn't have places to turn for their stress management needs, and they typically needed money, so they were really responding to our incentives as well. So we had something like a 97% retention rate on both the post fMRI session as well as the 4-month follow-up. So we got folks to stay in this trial. Okay, we're being reminded we have to stop at 2. Oh man, we have 3 minutes. Okay, I'll cut to the punchline. Punchline is that we changed the resting state signature in ways that we coupled their default mode network with regulatory areas of the prefrontal cortex. We saw connectivity with dorsolateral prefrontal cortex, both on the left and the right. And moreover, we find in the bottom right panel here that this change in connectivity mediated our improvements in inflammation. So our mindfulness program reduced inflammation, or at least a biomarker of inflammation, and at least a third of that effect could be explained by changes in this brain functional connectivity pattern. So I was going to mention another study in terms of going the other direction, in terms of how you might train the body with interventions in ways that might change the mind and brain. And certainly Kirk Erickson's been doing great work looking at aerobic exercise interventions. It's just one example of this, showing that he can modulate the actual volume of folks, of older adults' hippocampus in ways that may have benefits for memory and aging processes over time. So this is actually our last slide. So we've got 2 minutes. We wanted to sort of close with a couple of broader comments. Do you want to lead off?
Yeah, sure. I made the point earlier that this figure is an oversimplification of a lot of complicated, ideas that we're wrestling with. And one of the things, if you look at the bottom hand of this figure, we have health and illness as sort of lying along a continuum. And it's certainly not the case that these are sort of polar opposites. I think you can achieve well-being and states of health even though if you're suffering from a clinical or chronic illness. And that can play out in a lot of different ways. So we just wanted to acknowledge, some of the limitations and sort of arbitrary depictions of ideas that we're making in this figure, which raises, though, a lot of questions about the role of the brain in linking a lot of psychological, social, and other factors with health and illness. And there's certainly a lot of other things that we can talk about, but I think in the interest of time, if there are any last questions out there from the audience, we can field them and go a little bit over. Otherwise, yeah, we're happy to end things here. So we'll wait to see if any questions come in.
I do see a question coming in now, so just give me a moment and I can copy that over for you guys.
Doesn't look like we're
In fact, it looks like it's just a compliment saying, great job, this was excellent. But we do have a few moments for questions. If anyone has a remaining question, we can, stay over for just another minute.
Should we say a little bit about research challenges? Just because we've been sort of mentioning it.
Yeah, go ahead.
I mean, so one of the big challenges with doing health neuroscience research is that it does require you to bridge between domains, and certainly systems neuroscience approaches with social psychology and health psychology, methods and design. So there's certainly challenges there, but coming from a health psychology background, I sort of feel like we're built as a domain where we do collaborative research. And I think that while it can be a challenge or obstacle to sort of figure out ways to build in neuroscience pieces to your existing social or health psychology research, it may not be as hard as you think. In fact, building a collaboration with people who are doing good neuroscience at your institution is a great way to go. Sort of say, hey, we've got this paradigm that we reliably can show these effects on health. Would you want to work together? And that can really open up doors. And we're hopeful as people that have been trying to do this stuff that we see more research in this area. So.
Yeah, and even for graduate students and postdocs trying to pursue a co-mentoring model, if you're unable to work with like a health neuroscientist per se, having one mentor or advisor who is a health psychologist and another person who is a neuroscientist could work to your benefit in pursuing questions where you have to combine methods and concepts and so forth. So it can start early, in graduate school or as a postdoc, and then for junior faculty and others trying to pursue collaborative relationships I think is your best bet.
Yeah, I think for graduate students out there, I mean, Pete and I are both an example of this, where we sort of specialized in psychology or health psychology as PhD students, and then as we went on to postdocs, we tried to kind of create opportunities with folks doing neuroscience work. So I worked, with Matt Lieberman and others at UCLA, and you certainly came here and worked with Dick Jennings as a postdoc, and kind of used the postdoc period as a way to kind of build that toolbox out and protect some time to, ideally, do good stuff in the future. Yeah.
All right, so I'm just going to jump back in to wrap things up. So my name is Lisa Jaremka. I'm one of the co-organizers of these brown bags, along with Megan Robbins and Andrew Gears. We would like to collectively thank our two presenters for a really interesting and informative talk. Just also thank everyone on the webinar for attending. And as a quick reminder, all of our virtual brown bags are posted on our YouTube channel. You can find the link in the original announcement that you received for this webinar, and I also just posted a link in the chat window, so you should be able to see that. So thanks again to our presenters, and I hope everyone has a great day.
Thank you, everybody.
Thanks.
You. All right. This
Do we need to do anything?
I am trying to figure that out now.
Well, thanks a lot, Lisa. Thanks for organizing.
Yeah, thank you so much.